Bonjour tout le monde. Bienvenue à Paris. I'm Anna-Marie Wagner. It is absolutely wonderful to be here. I did iGEM the same year many of you were born, I found out today, which was great. And I was walking through the little kind of wall of history down there. If you look at I think it's called Food and Nutrition now. It used to be called Food and Energy. The 2008 iGEM team from Harvard won that, so woohoo. You all have done a lot more than we ever accomplished in our project. It's been inspiring and humbling to walk through the various sections this morning. So first of all, just congratulations to all of you for the work you've done over the past year and for being here and for advancing this field. I'm really, really, really proud to see what this community has become over the last over 15 years. This is going to be a panel about AI and AI's impact on synthetic biology and synthetic biology's impact on AI, which I'm actually much more interested in and excited about. I really don't want this to be just another panel about, is AI hype or is it real? And so we're going to try to get a little bit deeper. And one of the topics that I wanted to maybe just start with some reflections on is the culture of synthetic biology and the culture of computer science and AI. And I think this community in particular was really inspired in many of the early days by the computer science community, by the sort of engineerability of computer science, and by the potential to treat biology like an engineering discipline, right? A, C, T, and G, it's just like zeros and ones. If we can read it, we can write it, we can program it. I have been guilty of saying that many, many, many times. And while to some degree that's true, we all know that you can program something one day and program it the next, and it doesn't run the same way in your machine the way that if a computer scientist programs code on their machine, it runs the same way every time, predictably. Biology is complex. The machines are complex. We did not invent it, right? We like to say biology invented us. And so we're students of this incredibly complex machine. And so there's been this sort of tension between these cultures, this culture of computer science and engineerability and control and predictability, and the culture and reality of biology, which is sometimes random, and it sometimes doesn't work, and it's frustrating, and it's complex. But what's really exciting to me about AI today is that it is changing the culture of computer science, and it's making it just a little bit more organic, a little bit more biological. I'm sure, raise your hands if you've used AI. I assume it's 100% of you. ChatGPT, anything. If you put the same prompt into ChatGPT 1 second after the next, you will not get the same response. And no one really understands exactly how these systems work, why they say what they say. We can sort of explain the system, but we can't perfectly predict what it's going to do. And in many ways, that's its value. It's the lack of control, it's the lack of predictability that makes these systems interesting to us. And so one of the reasons I'm really excited to kick off this panel and to talk about AI as it relates to synthetic biology is I actually think we have this really exciting opportunity to bring these cultures together rather than trying to shift the culture of biology to look like Silicon Valley or try to force computer scientists to think like biologists, and rather than sort of fighting with each other, debating one another, really finding a new language and a new way to interact with these tools and with these disciplines in a much more productive way. So I'm really excited to kick off this panel. Joining me, I have Ahir Pushpanath. He's been leading biocatalyst development efforts over the past decade plus at places like Johnson Matthey, Basecamp Research, and now he's the technical director for biocatalysis at BitBiome, which interestingly has also built a really large genomic repository from some of its proprietary sequencing technologies. And so one of the things I'm excited to talk about in this panel, and actually all three of our panelists have a really interesting perspective on this, is the importance of data in AI and the tools that we develop to generate that data and use that data. To Ahir's left, I've got Elodie Pronier. Elodie leads biomedical discovery at Oken, right here in Paris, local here, and has been a key technical contributor to the creation of BioOptimus, which is working to create a universal biological foundation model in close partnership with Oken, and brings, again, a unique multimodal data set to that from its global hospital partnerships. And finally, we have Kelly Zalukuski. She's the Senior Director of Computational Neurobiology at Recursion, one of the leaders in this field, and she leads their London office. So in this role, she oversees, among other things, Recursion's partnership with Roche Genentech, which recently, and congratulations on this, delivered their first ever licensed phenomap. And so I'm curious to talk a little bit about what the phenomap is and their largest ever financial milestone. And I mention this just because one of the things I want to cover in this panel is also the business of this. You all are still in the very lucky and privileged position of being students. I really wish I was back there. But I assume many of you want to build your careers in this field. And so there is a reality around how do we make money doing this? How do we build sustainable companies that use these tools that you've been working on, that use AI to deliver real value in the world, not just scientific value, but also commercial value? And so maybe before we get to the big picture, let's start really practical. Because I think, especially for folks in this room, they want to think about, well, what can I do today? How do I even get started? It feels overwhelming sometimes. And so I'd love to just start the discussion with where you've seen the most practical value in AI today. Where you think it's actually making a real difference, and not just the sexy, hey, we're going to go design everything from scratch, zero shot with AI, but like, where is it actually making a difference in your day-to-day? Here, we can start with you.
Yeah, thanks, Anne-Marie, for that great introduction. First of all, I want to say, honored and humbled to be part of the iGEM. We were talking previously, all the students here are doing some crazy good science, and when we were in high school, I don't think we were at that level. So it's an honor to
when some of us were in grad school, I don't think we were at this level.
Absolutely. But yeah, talking about AI, I've always felt, with any new technology that comes on the horizon, human nature is to look at the dream state. What can we do with this new technology? It's when ChatGPT, as you mentioned, when it came up, everyone's like, oh, wow, this thing can do quite a lot of stuff. And then slowly you start seeing there's limitations to the technology, and then it finds its niche. And that's the same thing that's happening with AI, I think, in also the synthetic biology space. We have a dream state where we want it to be predictive. We want it to be able to give us innovation that we can't do as humans. But I feel like it's already found a niche state where it's only accelerating or giving us advantage in the mundane activities in the synthetic biology space anyway. So it's a good data analysis tool. It's a supportive tool. It's not near an actual innovation tool at the moment. That's where I think it is. But the greatest, the hope for the AI is for it to become a predictive tool for the design of organisms and enzymes.
Yeah, and to me, there is part that historically, AI has been already proving points in the chemistry space. I think it's the Nobel Prize recently showed us that indeed it has been accepted by the community. And to me, in biology, it is still to be proven where it can add the value. And I agree. I think the main subject is, okay, none of us, I guess, on the stage here think that AI is going to replace biologists, at least for the next few decades. And to me, it's more on supporting and accelerating what a biologist could do. And I think one good example that is becoming more and more clear is on the diagnostic. I think there is a lot of different companies that are really showing that AI can support pathologists and clinicians in their diagnosis, and especially in oncology. I think to me that's one area where currently I see a lot of impact of the AI.
And there's an entire diagnostics section over here leveraging synthetic biology, and it'll be really interesting to see over the next few years how AI starts playing into that. Kelly, where are you using it most today?
Yeah, I mean, I think there's an analogy to, this is my first time in Paris. God bless you all, I don't speak a lick of French. I'm that embarrassing American. And, I've already 5 times today pulled out my phone to say, oh goodness, how do you say this? Or to have it record and Google Translate tells me, oh, she meant this, right? And it's really effective for translating one spoken language to another spoken language. And I think in the same way, it's helping us translate in two different ways in tech bio. One of them is we create these massive data sets, so whole genome arrayed CRISPR knockout or whole transcriptome single cell sequencing knockout, and it helps us to translate that massive high-dimensional data set into something my feeble human brain can interpret, right? And the other one is, I think, a big issue that's sort of a little pain point that every tech bio company now has is like, we have biologists, we have machine learning scientists, we have data scientists, and they don't quite all speak the same language. But we're starting to develop these tools that allow our biologists and chemists to interact with these data sets directly using natural language. So instead of saying, hey, I have this kind of ill-formed question, and I'm going to go to my data scientist and frustrate them with my ill-formed question, they can say, hey, Lo, or hey, Gemini, show me the 10 most phenotypically similar genes to CDK12 and get that directly. And then they can visualize that all using natural language. And I think that's really going to accelerate that translation piece.
And I think that's also, and I'm sure the people in the room are going to be even more part of this transition, is somehow, as you said at the beginning, you have to accept as a biologist that even if not everything that you think as a biologist can be translated into a code version, is indeed making a step towards this thinking as a biologist, being like, okay, maybe there is some part of my reasoning as a biologist that actually can be transformed as a part of a code, not 100% of what I'm doing, but already if I'm getting a step there, that obviously I'm getting a step towards being able to talk and communicate the language of a computational biologist.
Yeah.
So, yeah.
I'd love to continue to pull on this thread a little bit, and Elodie and Kelly, you in particular, I think, work with both of those communities really very, very closely. And certainly at Ginkgo, for a while, and I'd say Ginkgo, was certainly leading the charge in terms of data-driven biological engineering. We were big believers in big data analysis tools to use that data. But even so, we've got a lot of really good biologists who, believe very firmly, like, none of this stuff matters unless it works in a lab, and your stuff isn't going to work in my lab. What have you found to be the best tools to start bridging that divide? You were just mentioning using some of these language models to help bridge it a bit. But I'm curious culturally what you've seen get the biologists on board and what you've seen really, I'd say, like, build respect and understanding among these groups of what the other brings and what the challenges are as you try to translate.
I think, at least to myself, I joined Oakin like 6 years ago, so I was the only biologist in the company for many years. And indeed, I did a lot of being a translator, so accepting to spend a lot of time with biologists, understanding exactly which was the easy part for me, obviously, because I'm a biologist by training. So it was, okay, discussing with clinicians, discussing with people in biology or clinics, understand what is their hope from AI, and often saying, like, okay, no, we're not there yet. Like, as we said at the beginning, like, it's not going to replace you 100%, so this is not possible from what you think from AI. This I know is possible, so let me talk then to the data scientist and explain what is your need so that you can translate it into a code. So that meant, to me, that's a lot of discussion and be a translator, being a bit of like your translator from the two team. And indeed, I think having more and more tools that help and interpret and push the understanding of the model that are interpretable by a biologist help open the door, the box, and it's not being a black box anymore. And then that helps also the communication because you have the same data that people can understand both ways.
Can I push back a little bit on this? So if tools are the way to communicate, doesn't that first have to start with ourselves as humans, that we want to communicate and be collaborative to begin with and use the tools? So it's something that I've had the luxury in my career and also at school, where my teacher told me, if you look at biology, it's actually just chemistry. And if the chemistry teacher says, actually, you know what, chemistry is just physics. So actually, everyone is looking at the other fields.
Hey, as a math major, I just want to point out physics is just math.
Well, there you go. So I think there's a human ego as well in every discipline. We think that that's the major discipline. Everything else has to come via us. So we have to bridge that gap first and say, it's happening at the intersection, first of all. So how do we get this communication piece to change mentality? Is it via the tool, or is it via some other medium?
I mean, I think what we're seeing when we build these maps of biology and then we ask people to suspend disbelief a little bit and like engage with what they predict, like the ideal outcome for us honestly looks a little bit like your ideal publication, where it's like, here's a thing you know already to be true, and here's an extra novel piece. And by showing them, hey, this does put out something that makes sense for a portion of it, they're more willing to take the leap on the things that are like truly novel, that they haven't seen before, that don't necessarily make sense. And so you have to help people bridge that gap between like, well, that's not in my benchmarking data set, that's not known ground truth, that's not, and like, well, but maybe it's real, right? And I think breadcrumbing them to that is kind of necessary, just because no matter what you're doing, the hardest part is still humans, right?
Yeah. That's a nice term you use, breadcrumbing, right? I think that's that's the way forward, spoon feeding.
Yeah. And Kelly, I know Recursion has obviously built a very large automated lab. I suspect part of the way you breadcrumb people along is you also do a lot of experimentation to prove that this stuff is actually driving value over time. As a framework for folks, I'm sure you've heard this before. When I think about like what drives value in AI, there's compute, which is what most people talk about, right? Bigger models, more powerful, yada, yada, yada. That was sort of the ChatGPT moment. There's technology, so new architectures, new algorithms. You could also call that talent, right? People who know how to build and use these systems. And then there's data, right? What is the data that we're able to train these models on? And there's sort of a reductionist view that AI is only as good as the data, and it's learning relationships between that data, but it really can't generalize beyond that. That's a debatable topic, just to be clear. But there's no question that data is key to building and scaling these models. And for a long time, I'd say in biology that has certainly been the biggest bottleneck and likely will continue to be the biggest bottleneck for some time. And so I'd like to have a conversation on the role of data, and maybe specifically how you see the role of data both on the pre-training side of AI models, so just building, maybe Elodie in the case of BioOptimist, building a foundation model for biology. Like, what does that mean? What is the data you're going to use? When will enough be enough? And then also on the sort of fine-tuning, reinforcement learning, validation side, how you see the role of data, kind of throughout the life cycle of AI. Kelly, if you want to start, just given Recursion's role.
Yeah, definitely. I think Recursion's point of view is a key differentiator that Recursion has is that we are building these scaled, fit-for-purpose, high-quality, consistent data sets in-house. And so the main technology that Recursion started with 10 years ago was cell painting. We do a massive amount of high-content imaging. And honestly, the first half-decade of Recursion's existence was doing all of these, call them boring things, but it was like the control strategy, the standardization, the automation with all of the robotics in the lab. Like, how do you guarantee that an image we took in 2015 we can directly compare to an image we will take in 2025? Right? That's, anyone who's worked in a lab knows that that's like a very, very hard thing to do, because often you can't compare an image you took on Monday to an image you took on Wednesday, right? So a 10-year gap is really something. So I think really putting in the effort in the ground stage to create the data you need to meaningfully deploy AI against biology is one thing. And then to connect it to our previous conversation, if we get a prediction out of our maps of biology, the very next thing we do is say, oh, okay, how can we orthogonally validate that? Like, what is the next assay we can use to help prove out that, like, hey, that thing we're seeing in our map is likely to be true, or we're convincing ourselves and we're convincing our partners. And a big part of the Recursion philosophy on doing this is that we're trying to collect all of these high-dimensional data sets in a way that they can be fed back into the beginning. So we create these virtuous cycles. So like any data point we collect for a specific program feeds back into the original training data set, and it's available to every future program we will generate. And I think that is going to help. That closed lab in the loop piece is like a key part of the philosophy of Recursion as well.
And I think it's interesting because at Okin we took exactly the opposite vision, which comes with the same limitation, but on the other side. So the way we are building the foundation model is because we think that we want to treat humans. So the best way to treat human is to find human data. So indeed, we don't have a lot of data in-house currently, but we're looking and working a lot with academic partners, people from clinicians, people in the real world to get access to their data, and that's how we build the models. Like having a lot of different type of data, so multimodalities, on a lot of different patients, so that we hope, and again, it's a hope, it's being tested currently, that you have less bias in the way you develop your models because they have seen more type of patient and more type of data. But then so that, on principle, is amazing because then you force the model not to be so overfitting in one population. But then you have to validate the results of your finding, and then you go back to a lab and you go back to a petri dish, which you know is not representative of your population of interest from the beginning. And then what do? If you validate your finding, then happy ending, happy story. If you don't validate, who is wrong? Is it the AI or is it your petri dish? And right now, it's really, really a complex question for us.
Yeah, and I think you're both operating in this incredibly kind of difficult field where we just don't have models, whether you're talking about a computational model or an animal model, that are reliably translatable to whatever that next step is, right? The computational model is so-so when you get to a petri dish, which is so-so when you get to an animal, which is so-so when you get to a human, and so you have all these different translation steps that you're trying to bridge. Is it easier in biocatalysis?
I was just going to say, maybe it's a little easier because we don't have to deal with mouse and things like that, but I think the lab scale results you get with an industrial enzyme is very different to the enzyme performance in a reactor at process scale. So we do still have that same problem with data. If you're going to find data, do you use lab data to train all your models, or do you use process data from actual plants, right? So I agree with both of you in the way that lab in the loop is a must also for industrial biotechnology. If you're going to be developing any enzymes that work in a process, you've got to validate it at some point. And data for me is not just for AI, but data is the only thing the customers trust ultimately. They're not going to trust your hypothesis on AI. They're going to be trusting, does it work, right? Your final product of AI. So for me, I'm always looking at data. Can we get the right data, which the customer is convinced with? Is that the right data then to train the AI model so we can give more solutions to customers? That's how I'm viewing the whole data aspect in AI.
How do you all think about, you just mentioned data is the only thing, lab data is the only thing that customers trust. I think when folks talk about AI in biotech and synbio, they're typically focused on the discovery side, at least this type of community is. So we're going to discover a novel target, we're going to discover a novel biomarker, we're going to discover a novel therapeutic. And some of the pushback I've heard recently from the broader community is, yeah, but that's not actually the problem. Hypothesis generation isn't the problem. Hypothesis testing is a problem, and what hypothesis testing really means is you stick it in a giant bioreactor. You stick it in a human in a clinical trial, and that is the bottleneck. And so I'm curious how you see the space evolving as we get a lot better at hypothesis generation, and I think that's already happening. I'm sure, at Recursion you've got 100x, 1,000x more great ideas than you can actually test in a lab, much less a clinical trial. And so how are you thinking about the ecosystem evolution, given that we have this massive influx of design and hypothesis, and we're still bottlenecked at that very last stage?
Yeah, that's a very good one. I think that's exactly where automation comes into play as well. So the way you collect data has to be at the same pace that AI is craving the data. So this is a really important thing for the lab in the loop. The way you collect the data has to be really, really fast-paced. It has to be cheap as well, and the types of data you collect needs to be reflective of the customer problem. So those, I feel, are the major limitations of data collection in general for AI. And if the tools and the engineering principles don't catch up and automation doesn't catch up, AI is always going to be in the state of, we need better data, we need better data. So that's where I'm at.
But to me, one of the main limitations of this is obviously going to be money.
Yeah.
Because if in an ideal world, I would love AI to create indeed thousands of hypotheses a year, a second, a minute, you tell me, and then to test them in the lab. So I guess indeed automation is one step, but I don't know if in this room or ever I would...
Anyone have money?
You have billions of viewers to spend on this, because to me that's the dream I have, which is, okay, I have thousands of hypotheses, I have hundreds of models on which I can test my hypothesis and not rely only on one model, and then I will have the ground truth that we are all looking about. But when I'm thinking of this, then who does the check? I haven't found that person yet, so...
Yeah, I mean, one thing is, I think you're totally right. Like, it's thinking about how do you miniaturize, automate, industrialize, scale that next step always. And so like recursion always starts with high content imaging, but like we've also collected more than 1 million transcriptomes, right? In 1536 well plates, compounds, like miniaturizing that process has helped us to collect data at scale on the next validation step. But I think another way we can think about it is like AI can help point us to the most fruitful places to do that testing. Like, can we intelligently deploy active learning to say, oh, well, what is the chemistry library we should create to most efficiently explore the space, given that there's more possible chemical structures than there are stars in the universe, right? We can't possibly scan them all. Like, can AI point us in the direction of like, okay, this is where you would start, and then this is the most fruitful direction, and to do that sort of like binary search more quickly, I think is another place where it's going to help speed up that development process.
You all are talking a lot about data generation upfront as part of your process validation. How have you balanced the different types of data you could generate? There's data you generate for a customer program, right? Hey, I need to fine-tune my model in this particular class of enzymes, or I need to go do a bunch of validation work, versus I think my model is hungry for this type of data. Like, I think it'll just get better. I don't really know exactly how or why, but it seems hungry for that. Like, how are you thinking about what I would maybe describe as like proactive data generation and more generalized data generation, or more, maybe you could call it like unbiased, versus focused on an individual program?
Yeah, I think there's a big hypothesis in the field of enzyme design anyway, that if you really have the biggest data set possible of natural enzymes, then AI is somehow going to learn the ground truth of what it means to be a functional natural enzyme. So that kind of data is more not led by the lab, but it's for generative AI, surely. So here, collecting more natural data is what BitBiome does, and we built this huge 2 billion sequence database of microbial diversity. And the hope there is maybe we feed it enough diversity, AI will finally find the ground truth of what it means to make a natural enzyme. But is that going to be enough then to make an industrial enzyme? That one is not represented in natural enzymes. So you have to have the lab in the loop for that kind of data. So both is necessary, I feel. You need to have that vision of let's just feed it more data, and at some point you'll get some answer. And you also need, I'm going to kind of navigate it to this particular process-ready enzyme state. So that's where I am on that. Yeah.
I think so at Recursion, a thing that we're doing, my first year at Recursion, we all got a box of gifts for Christmas, and one of them was the book Loon Shots, which is sort of centered on the idea that like ideas that are worth chasing, at the beginning people are going to say, you're crazy, that's never going to work. And then some sort of process ways to go about like, how do you do this sustainably, chasing loon shots? And one of the key themes in the book was like, okay, you're going to meaningfully wall off in your company the artists from the soldiers. And so we have a whole department at Recursion, actually a large fraction of the company, that's dedicated to building and maintaining the Recursion operating system, the Recursion OS, which is like proactively generating more maps in new cell types, proactively developing more powerful models to just like keep building what we're doing at Recursion to serve all of our partnerships and all of our programs. And then we sort of wall off our machine learning researchers or our people who are looking at these like longer term, this isn't going to read out for 3 years or 5 years, but if it works, it will be game changing. And so we sort of operationally make sure that that happens.
Well, us, it's a bit of more a testing and see if we're happy kind of approach, meaning that obviously a lot of data scientists that are coming at Oakin, they are really good at using images as a data modality to make any predictions. So obviously we have, and even on Bioptimus, the foundation model, they are working amazingly well on image-based prediction. So that's an easy avenue that was for us to take. And then on the omics data, to me, it's not as striking as I said at the beginning, like where AI is going to be able to change to bring the gap or to change something. And so as we think that the more the merrier, because obviously then you can grasp better the heterogeneity of the disease. Is that going to be the best suitable approach for AI? I'll come back in 2 years and I'll tell you.
I like this incremental approach. Like one of the questions I get asked a lot is like, okay, but like how much data do you need? And you mentioned money earlier. Mm-hmm. One of the groups I work with is DARPA, the United States kind of Defense Department's research group, and they want to know this question too. Like, they've got money, they're willing to spend the money. They want to know, what should we spend it on? Like, how do we know that if we go put hundreds of millions, billions of dollars to work to generate the data that you all in the community want, how do we know that's going to make a difference? How do we know that's going to be enough? And so you've mentioned kind of these little hypotheses that you're testing. How do you all start getting conviction around, hey, I think investing in this area of data generation, because the reality is, if data generation is the bottleneck, data generation is also really expensive, and you have to therefore have real conviction before you spend limited resources on that. How do you build that conviction, and how would you help the people who do have the money, build the conviction to start putting it where you want it?
So us, we have done a lot of testing on because obviously people always tell you what is the minimum that you need, because it's always better to start with the minimum. So we did a lot of testing on every not every, but the majority of the predictive models that we have at Oaken and by Optimus, we first kind of synthetic, in a computer, you can kind of see the impact of the numbers on the performances of your model and say okay, after this bar, I know that the learning and the performances of the model are really bad, so that's the minimum that we usually require. And obviously you can do that by modality, or even if you have different approaches and type of data to kind of say what is your minimum. And then there's amazing people that indeed, even when they have complex model, they are trying to also see what was the weight of every independent modality in the performance of the model. So if you lose one modality, what is the impact on the performance of the model? So I'm not saying I have an answer that is always clear when one customer is coming to me okay, I want to predict patient survival. How many data and what type of data do I need? I don't have a clear answer, but I start to have an idea of what is at least the minimum required. Then, is the minimum sufficient, or do I want thousands more? It's still a debate for me.
I have two things to say about this. One of them is, at Recursion, I think we find we have this milk and cookies problem, which is you finish your cookie, you still have a little more milk. Oh, well, I guess I need another cookie. Oh, no, now I need more milk. Oh, no, I need... And what we're seeing is we get to be really actually kind of data rich and then compute limited. And then we invest very heavily in compute. So recently, I think over the summer, we rolled out what is now the largest in biopharma compute cluster. And by deploying our newest foundation model on that compute cluster, we improved recall in our benchmarks by 25% in 2 weeks. So clearly we were compute limited, right? And now we're back to saying, okay, well, now we have enough compute. What data should we generate? But I think the other place we're really thinking a lot is around benchmarking and what does good look like in this space, given that you're trying to discover the unknown and find novel things. And so Valence Labs at Recursion, which is this machine learning research arm, has recently started this collaboration. I think on the initial publication were J&J, Bayer, AstraZeneca, and Recursion, thinking about okay, how can we all come together to say these are going to be the benchmarking data sets, these are going to be the sort of ground truths, this is the MNIST data set for TechBio, right? When we're testing these models together, can we all sort of come to a consensus around how we will test them and how we will decide what good looks like? Then we can say oh, how are we doing against these benchmarks we all understand?
I think, Anne-Marie, if the question is more around how do we get the investors to put money into getting these data sets, I think it's already happening. A lot of startups are still raising pretty substantial amounts, even in this bad climate of investors. So they're managing to convince the people with money that there is a promise of AI. Yeah. But are they convincing them about exactly the data sets required to pull it off? No. So that's the issue. I think when you get to the honesty, I don't think a lot of us, even scientists, we have to be a bit humble here. Do we really understand biology enough to know exactly what data set to collect? So you're making a gamble there, first of all, on what kind of data set we need, right? It is. And then once you've gambled and you've taken that leap, you bring all the millions into it, and in halfway you find it's not working, then that's on you, right? So that fear is there for the people asking for the money as well, so we don't talk about it. We talk about only the promise and premise. So I think there's a thing that we can learn about asking for money from people that do have it. Maybe we just need to be a bit more honest at the beginning, so they give that minimum, and they don't give too much, and then you're scrambling, trying to make up the profit. So that could be it.
Yeah, and to me, because there is days, I'm not going to lie, at Okin, where we have amazing data and we're trying AI, and it fails. That's the reality, right? But I'm always thinking that, okay, even if you don't do fancy AI, we have data that was not existing before, and you do computational biology, and you find other answers that are not for AI, but are more for even a biologist like me, like more ground finding. And to me, that's still like a really important step towards understanding the biology, understanding the disease that we're working on. So I agree. Sometimes I'm like, yeah, we're gonna go skyrocket with AI, and I'm like, okay, it doesn't work. Okay, still not useless because we can use those data for something.
Spoken like a true scientist.
Trying to be.
Although I will put in a small plug for a topic I've gotten really interested in, which is, and there's sort of, some of what you've said, Kelly, there's kind of the unsexy blocking and tackling side of it, and then there's the really exciting kind of autonomous science vision. And one of the really interesting foundational research problems in that is, can a model develop the experiment to test its hypothesis? And if one of those hypotheses or one of those goals of the model itself is, I want to get a lot better at developing hypotheses in a particular area, then you start having a partner in these models that can help you determine where to go generate data. And I think that'll be really exciting. We've got just under 10 minutes left, and I want to chat a little bit about the business model. And we've started talking about money and investors, and so we're sort of naturally getting into this space. I think we've already seen, and this is certainly true in pharma. I think it's probably also true in industrial biotech to some degree, but it's very stark in pharma, where you used to see these fully vertically integrated pharmaceutical companies that did everything from drug design to development to distribution and marketing. And so they owned that entire value chain. And now, well over half of drugs that go into a pharmaceutical company's pipeline is partnered or acquired from a smaller, more innovative biotech. And obviously, Recursion's business model is doing a lot of that development, such that can then partner with pharmaceutical companies. And so, in many ways, the role of pharma has gotten squeezed into this much smaller piece around late-stage development and then distribution and marketing. As AI, comes more into the fold on discovery, is there a risk that kind of more traditional biotech gets squeezed in the middle, where we're not doing, distribution and commercialization, and now suddenly one-person companies run by the folks in this room in high school can do really amazing design work? Are we stuck in that translation phase, or how do you think the business model evolves in this field as AI develops?
It's a tough question. I think in the pharma space anyway, it would be a great reality when anyone in this room could make their own multi-billion-dollar pharma. That's what I want to see. Why is it only a couple of 15, 20 handful of pharmas running the game, when really AI has empowered all of us to essentially become our own innovator and own distributor? But it's a hard game, right? So that's the thing. You need the money, as we go to before, and you need to have the proof points. But the way I see it, the business models for the small fish at the moment is still reliant on the big players to take a gamble, and then at some point the big players buy them out. Or you take the risk and say, I believe so much that I can go to market with my own therapeutic. I don't need a big player to support me here. But that requires your investors to take the risk with you. So that, I feel, it's a gamble again at the very beginning of what kind of model you want to follow. And at some point you're going to have half-half. You're going to have to be in bed with one of the big players as well.
Yeah, I mean, at Recursion, we have a number of different irons in the fire. I think our main thing that everyone knows us for is developing drugs and developing drug discovery programs and partnering those out with large partners. But we're also doing things like we are partnered with NVIDIA, and we've put some of our models on BioNeMo so that other people, even you guys, could access those models and work with them. Or another thing we're doing is taking the large language model tools I was talking about earlier. One of them we built internally is called LOW. And we are using sort of an app store model where you're creating the ecosystem and you have the content. And so Bayer is the first company that's working with LOW, but we're allowing them to use these large language model enabled tools to directly query Recursion data. And so it's oh, you want to get insights into Recursion data? Well, here's the app you're going to use to do that, and hopefully by that you're sort of capturing some of that space.
And to me, it's really complex because, I think I've seen a bit all, because Bioptimus is obviously a tech company, so we are more talking about delivering a platform that is indeed hopefully delivering to the world really good algorithm. Is that enough as a business? I don't know. I think right now it is indeed. Because AI is the hype, it's really interesting to investors. But then when you pass the phase of investor, what happened to us? I don't know. Which was the beginning of Okin as well, where we started really as a tech. And then you go to the big players, like the happy few, that has the money to kind of bet on your tech and your platform. But then either your tech is working really well and indeed they buy yourself, so which means that it's a bit the end of your own story, or they are I like your idea and I'm going to try to do it myself, which is obviously then their bet to kind of good luck to kind of replace a company like us that are really focused on developing AI. Or you take the bet of why can't I become myself one of the big players that I'm founding my own targets and I go to the drug development. But that's then balancing the investment that is coming from tech people that are the ones that usually invest in you at the beginning versus having enough money to fight for the 15 years that it's going to take you to have the proof point that you have an asset that indeed was successful in clinical trial. To me, there is a bubble in between that is really the challenge to a lot of companies like us for the moment.
I mean, supposing any of our technologies we're developing suddenly becomes, and you have a eureka moment and oh my God, this is actually working, will you take the gamble then and say, you know what, I'm going to make my own thing. I'm going to be making products with my technology rather than selling it on? Is that what we're all waiting for, to have that breakthrough moment?
Well, I think I think this actually reflects a little bit of the bias in our industry, where right now the only way to prove that your technology has any value is to make an asset that is valuable with it.
Yeah.
And the unfortunate side effect of that model is that there are very few, if any, businesses in our space that have sustained R&D, have sustained innovation after they made a product.
Yeah.
Either the product fails and everyone's like, oh, well, the technology must have been garbage because the product didn't work, or the asset is wildly successful, gets acquired by pharma, and the R&D team still doesn't have a job to do, because now the only thing anyone cares about is that drug, because that's where all the value is. And I'll say, for me, one of the things I'm actually most excited about and optimistic about is the potential of AI to allow biotech, tech bio, synbio, whatever we want to call it, companies to monetize the R&D process itself, so that innovation engine can in itself be a sustained business model and isn't only reliant on those assets. Obviously, the assets will still be maybe the most valuable piece, but is there a way to at least sustain that innovation engine because AI can help make some of this technology more translatable? We only have a couple minutes left. I want to be cognizant of our audience here. These are all college students and even high school students who are building their studies and probably their careers in this space. I'd love to just go around and get a 30-second kind of piece of advice from you on for this group.
Yeah, I mean, when I started grad school at Stanford, I was coming from having done college in Arkansas, which is a place that's not particularly known for being really tech forward, and I landed at Stanford and I was like, I don't understand why anyone would do computer science. My roommate was a math major, and when she said for loop for the first time, I imagined a clover leaf. No idea. None. But once you see that oh, that's the water we swim in, that's what you're going to need in order to be successful here, I went and started learning those things, and now, this is what I'm doing. I'm a major leader at a leading tech bio company on the bleeding edge of things. And so I think my call would say don't be intimidated by what you don't know. A lot of these things are really less difficult than you make them out to be in the beginning when you know nothing and it seems intimidating. Just getting that first step in lets you know that you can do all of these things.
It's also believe on thinking outside of the box is really important, because I already knew I wanted to do biology, but so I did a PhD and then I did a postdoc and then I wanted to have my own lab and blah, blah, blah. This kind of like academic career path that everyone tells you when you're starting. I mean, when I started at the time. And when I joined Okin, I was like, it was a small company of 40 people. I was the only biologist full of like data scientists that I remember the first lab meeting I went to, it was full of mathematic function and I was like, whoa, that's a huge gap from being a lab rat, right? And so to me at the first I was like, what bad can happen? Like, I'm going to have an experience, and if it fails, I'm going to still open the door to something that I was not thinking I was capable of. And here I am 6 years after. Now I understand what AI is, I understand the math function, and I'm even able to talk about it. So I think, trust in yourself and saying like, okay, it's not exactly the path that was supposed to be mine, but let's see where it goes.
Yeah, I think both of those are very good. I'll just add that we're living in the information age. When we were in school and, I guess information was there, but not at the level it is at the moment. So as you in high school and undergrad, PhDs, you're all fully saturated with information, and there's so many worldwide issues that are happening. You feel stressed that you have to be, we are relying on you to solve the next problems of the world. So that pressure sometimes can distract you from doing the scientific rigor that's needed to achieve. So I just follow your heart and go one step at a time. Don't get over-pressured into delivering the future solutions.
Yeah. Thank you all so much for joining. The only thing I would add is become bilingual or trilingual. Like, learn computer science, learn biology, learn business so that you can help be a translator, just as all these folks have become translators in their industry. Congratulations again. I'm so proud of all of you, and thanks so much for sticking with us. Have a good one.
Thank you.
This episode of Cell and Gene, the podcast, is brought to you in partnership with Thermo Fisher Scientific. Thermo Fisher's cell therapy processing instruments are designed to help customers transition from process development to commercial manufacturing, utilized as standalone devices or integrated as part of a closed modular process. Thermo Fisher Scientific recommends Gibco CTS DynaSelect Magnetic Separation System, which is a next-gen cell isolation and activation instrument. Gibco's CTS Xenon Electroporation System allows customers full control to optimize for a variety of cell types and payloads. And Gibco's CTS Rotea Counterflow Centrifugation System is a closed cell processing system supporting a broad range of protocols for cell separation, washing, and concentration. Customers can rely on and streamline their drug development process with Applied Biosystems QualTrack qPCR and dPCR quality control tools for robust and reliable genetic analysis across various phases of drug development, supported by relevant, compliant documentation. Hello, listeners, and welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and my guest for this episode is Shape Therapeutics co-founder, president, and CEO, Dr. Francois Vigneault. Francois, thank you for your time, and let's jump right in. So, Francois, briefly talk us through Shape Therapeutics and the company's mission.
Yeah, thanks for having us. The aspiration why we started Shape a couple of years ago was really looking forward into the next couple of decades. What would be the technology that would probably impact patients' life the most? And to us, the two key things that became obvious was AI, application of AI and large data set. And now it's hot, so I feel I'm chasing, the buzz of now. But 5 years ago, we were telling that story. And it's using AI at a scale that people haven't done in life science at the time. And the other technology was RNA. And now people know of RNA because of COVID. But the idea that, RNA is a 4-letter code, would make it very amenable to computer science. And merging these two fields together, we call that programmable RNA medicine. And the dream is to be able to design, therapeutic cure for patient with disease with unmet need using RNA-based technology, but do so using the power of AI to shortcut, development of R&D and reduce costs and build drugs that could work for a single patient that has its own unique disease in the world, or for disease that, 50 million people have, and find a way to make this cost efficient for society.
Good. Okay. And I'm really glad you brought up AI. We're going to get to that in just a little bit. But before we do, I want to talk a little bit about, so you mentioned, Shape Therapeutics develops RNA technologies, and you're shaping the future of gene therapy. Perfect. What exactly is in development?
Yeah, so the first, let me explain what kind of RNA-based technology we're building. People are familiar with COVID. You give a messenger RNA that codes for the spike protein. You give that using LNP into a vaccine and mix the protein. So what we do at Shape is quite different. What we found was there's a set of protein in you that is able to edit your RNA. There's a protein called ADAR that exists in all of your cells that edits your RNA at hundreds of thousands of locations. So in your genome, you have about 20,000 genes. They code for messenger RNA. Those messenger RNA then brings the instruction to make a protein, and protein is the function. So when you have a disease, there's three paths to try to treat the disease. Either you fix the source code at the DNA level, either you make an antibody or small molecule that historically tackles the protein side. So we're operating in between the two. We're trying to find a way, can we just do a software update on your RNA as opposed to change your source code or give you a medication that you have to constantly take forever? Could you, basically the software dispatch of the RNA? So this protein ADAR is editing your RNA as we speak in hundreds of thousands of locations in all of your cells, specifically strong in the brain. So what we did is we decided to study how this ADAR protein is recruited to the natural site where it's doing its job. But instead, if we could provide a slightly different RNA instruction, could we hijack this ADAR protein and bring it to a chosen location to make an edit and correct a genetic disorder? And I'll give you an example. For example, in Parkinson's disorder, there's a mutation that some patients have that's on the gene LRRK2. And these patients that have this mutation have a very severe onset of Parkinson's disorder. So the idea is if I can provide a small RNA to the neurons of this patient, I can bring that ADAR protein at that location and fix the mutation that they have there to return them to a normal, sequence, and therefore prevent or stop the progression of Parkinson's disorder. So that's one disease that we're actively working on. The rest of the Parkinson population also overexpress a protein called alpha-synuclein, and this creates some of the Lewy body that are toxic in your brain by creating aggregate. So in this case, they don't have a mutation in the gene alpha-synuclein. But what we can do with this tool is we can still go with a small RNA and recruit ADAR to shut down the translation of RNA into protein and reduce down that alpha-synuclein to a wild type level. And we can completely control if you want to bring it to 50% or 70% down or close to zero, which is a brilliant application of RNA tool. So in contrast, so we're operating in the editing space. So people have heard of CRISPR or base editing and tools like that. These tools are meant to edit DNA, where we're tackling editing at the RNA level, and we think there's many, many benefits to do that as opposed to DNA editing. So we're in Alzheimer, we're in Parkinson's, we're in a disease called Rett syndrome, we're in some muscle disorder, we are in liver disorder as well, but we're definitely heavily focused on CNS. CNS seems to be a sweet spot where there's a ton of disease that have no treatment, yet the ADAR function is very strong in the brain. So we're able to capitalize on that and try to build the right treatment for these class of diseases.
Good. Okay, thank you. I want to talk about artificial intelligence broadly and in general as it applies to cell and gene therapy in the sector at large. So AI is a major topic in the space. What, from your perspective, what is the potential of AI?
Yeah, that's a big question. It's a short question, but it's a big one. Ultimately, I think now we're seeing quite a shift in the application of AI in recent months. some people are crying doomsday that AI will take over, but you could also look at AI as what if you could use AI to build drugs very efficiently, right? And I'll give you an example where I think where this is going. I believe that probably in the next decade you'll be able to pretty much develop any drugs using just AI and have your winning candidate ready to move forward in the clinic. And that's going to be across small molecule, antibody, and RNA-based technology as well. And we're already doing that at Shape to some degree. So what we did initially is build massive training sets. For your AI to work, you need to give it data. And if your data is junk, you won't get anything good out of it. So we ended up designing millions of these guide RNA. So the thesis was RNAs for ribonucleotides. So instead of classic biology where you choose which part you mutate to get your action, we said, well, what if you were to make every possible permutation? If you did that, you'd be querying the finite space of all solution, and you'd learn what is the limit of the system. Sadly, it's too big, right? So if I have 100 nucleotide long RNA, that's like 4 to the power. It's 4 to the power of 100. It's just too big. So what we do is we go as big as we can. We inspire the structure from nature, and we go as large as we can. And this is where AI comes in. You get multiple positive hits, and you get many more negative hits. So you may not have found your best winner, but if you find enough in a cluster, you could start studying patterns and see what made these work versus the other one that didn't work. And this is where machine learning becomes quite powerful. Historically, people would use more regression model and Bayesian algorithm to study using machine learning to find why can it didn't work. Lately, what we've seen is the application of generative AI. And it's not quite made its way in life science just yet, but we've been dabbing with that quite a bit recently at Shape for the past year. So what we've been doing is akin to, a Midjourney or a DALL-E, these image generator that you see online now, where you say, hey, paint me a unicorn on a tricycle, and it does it in a minute, right? And it's just brilliant. So the way it does it essentially average, what, a unicorn should look like and slowly make it more precise. So we started thinking, can we do that with life science data? Can we do that with viral engineering, or could we do that with RNA? And we took our training sets of all these millions of RNA that we've actually made in the lab and tested to get all these learnings, and we plugged it into generative AI algorithm. So we had to tell, the machine learning model that, what is a good RNA and what is not a good RNA. But instead of doing the classic machine learning model that you've seen in life science, we started saying, hey, let's pretend these are painting, the same way that you're painting a unicorn. Pretend these RNA are a painting and just paint me the correct RNA for my problem based on that. Same with viral engineering. We had to find a way to somewhat pixelate into an image the value of an amino acid and say, hey, this is what an apple should look like. So draw me an apple, but essentially it's drawing a virus. And it works. It works. What we got out of this is guide RNA that are much superior to anything we've built in the lab in the past 3 years in record time, right? So we also face solution problem where we couldn't find a perfect solution, where we needed the 3 locations, for example, in Alzheimer's disease. And in the lab, we found solution that can edit position 1 and 2, or 2 and 3, or 1 and 3, but never all 3, never the right way. This is exactly the kind of data sets that you want to apply to machine learning. And what we got out of it is actually guide RNA that is able to efficiently edit all 3 positions perfectly in records time without having to be in the lab. So now we're at the point where when a pharma partner comes to us and say, here's my favorite disease, here's the mRNA I'd like to tweak, we don't have to go in the lab. We can essentially just plug in the computer, spit out a top 10 list of RNA candidate, where historically we'd have to spend a year and a half like screening in cells and so on. So we get a top 10 list of candidate, then we go in the lab and then we validate them and then we mature them further and we take this learning back to the machine learning model. So I think this is happening in all field of life science, from small molecule discovery to antibody folding. If we look at the work at AlphaFold, it's quite exciting work there, and to what we're doing at Shape. And on the RNA front. But we are definitely pushing the boundary of AI and RNA. We're applying ChatGPT-like process to life science data. And it works. It works really, really, really well.
Yeah, that's actually exactly where I wanted to go. And I'm glad you said this is what we're doing at Shape, because that's what I wanted to ask you next. Okay, so how then is Shape Therapeutics leveraging AI with the goal of developing these technologies? Where are you guys at?
Well, we like a hard problem. So, I think most investors would say, don't be an Alzheimer. You're going to break your face. Everybody fails there. But historically, I like to work on the hardest thing, because if you solve that one, then everything gets easier from there. And life's short, so may as well do something, useful so you can sleep well at night. And if you fail, it was worth trying. I think tackling CNS disorder out of the gate is quite challenging. Going in Alzheimer's and Parkinson's is, these diseases are poorly defined, the cause is poorly defined, but there's enough hint that there's a path to take there. And I'll tell you the story of Alzheimer's, because it's a compelling one. There's antibodies that have been approved now on the market. So Alzheimer's, I should be careful my words, not necessarily a proven cause, but there's a gene called APP that's expressed in all of your cells. And APP has a normal function that you must have to be alive. The problem that we see in Alzheimer's patients is there's an enzyme called beta secretase that cuts APP, and these fragments aggregates and become toxic to your neurons. All the antibody, all the small molecule, everything that's ever been developed in the 150 clinical trials that's ever happened in Alzheimer's have almost pretty much failed. And the reason is very simple. As soon as they start seeing efficacy, even in the antibody that are approved on the market that are quite controversial, you're starting to flirt with toxicity, because you cannot get rid of APP or beta secretase or their pathway. So our thesis was, is there a way to prevent aggregate formation without suffering any of the toxic consequence of getting rid of one of these natural enzymes? So we turned out to population genetics for that. So in Iceland, people have a normal variation called SNPs. They have SNPs variation on APP that makes them protected from these pathogenic cuts. So they have a normally functional APP like you and I, except the sequence at the location is a small variation that they have in their genome. Doesn't impact APP, but what it did is it completely prevent these enzymes from cutting at that location and creating these pathogenic aggregate that seem to be destroying your brain. This is a natural thing happening to them. They're quite healthy. They actually have no onset of dementia, Parkinson, Alzheimer's as they grow in that population. So we said, what if I could turn you and I into an Icelandic, APP person, right? Why not? Because we know it's safe. They have it. But could we make tool to do this? So this is impossible with antibody or small molecule. You can't change the code, right? You could also hardly do it with CRISPR-based tool, because you can't get efficient editing in neurons with these tools in vivo in patient. And this is where RNA editing is quite exciting, because we're talking, again, just a software patch. If I could deliver a small RNA and it goes to your neurons and it essentially encodes ADAR to change that sequence of RNA into an Icelandic-like sequence, boom, I'm preventing, cutting these APP into beta secretase. And I could potentially completely prevent or stop the progression of Alzheimer's disease. So it's a super high risk, high reward project. We're years away from the clinic, but the concept is so good that we have to try. We have to push these things forward. And this is the example where we had to make 3 edits at one location, which seemed intractable at the time, where AI came in and essentially solved it for us. We gave them very good training set how to fix portion of it in a different way. And then we told the algorithm, find me the perfect solution. And what we got out of that is guide that completely works. And now we're pushing them the next step and eventually in the animal model.
Excellent.
I don't know if I completely dodged your question with this. I went on the road, but I felt a good use of connecting how you make great data set in AI. Because I think what we're going to see is a lot of AI company or machine learning company will say, oh, I'll do drug development. It's not that simple.
Mm-hmm.
Most if I go at Microsoft down the street, they have software engineering, machine learning that are way better than what we have at Shape, I'm sure. But they don't have the ability of building the right biological training set. They just don't have that. They don't have the sequencer. They don't have the knowledge of microbiology. They could acquire it, but you have to hire these people with PhD that have worked in the lab for quite a while, because you're only as good as your training set. And most life science companies don't have enough machine learning or AI knowledge to really push the boundary the way that an open AI company is doing right now. So we're trying to find the intersection of the two fields. And one way we do it is by talking to these companies and trying to, leverage ground of collaboration.
Perfect. Okay. And your response was wonderful. That's just.
I tend to do that. Yeah. It's worse when I drink red wine, then it feels like.
I think that goes for everyone. No, thank you. That was perfect. So I actually do want to better understand, if we're talking, without being a futurist, but with, if we're looking ahead 5 years, talk us through what you think the intersection of RNA, AI, and synthetic biology might help evolve the gene therapy space in, a handful of years.
Yeah, great question. The problem with any gene therapy ultimately comes down to delivery, right? So at Shape, we're building these, RNA payload. We call it RNA fix. Even if I was to develop this perfect RNA guide for Alzheimer's disease, there's still a challenge of, can you put in the right cells? And that's what's been hampering life science, gene therapy field in general, is getting the right, payload into the right cells. Another company have different payload and all have value and different purposes. You spoke with company that do gene replacement, for example. You will be speaking with people that do vectorized antibody. At the end of the day, can you put in the neurons or can you put it in the lung? So the tool that's quite popular is using AAVs, adeno-associated virus. They're a naturally occurring virus that don't have a lot of symptom in human. So and they're fairly immunogenic. They can stick around long enough to deliver their payload before the immune system, takes care of them. So that's the tool that's been used in gene therapy for the past, 20 years to try to deliver these payload to the brain or the heart or muscle. Sarepta just got approved their gene therapy in Duchenne muscular dystrophy, so that's fantastic for the patient. So they need to deliver their payload, which is microdystrophin that expresses this protein in muscle, heart, and diaphragm. The challenge is AV go 99% to your liver and only 1% elsewhere, less than 1% elsewhere. So what the industry has done to compensate for that is give a higher dose. Right? But when you get a higher dose, then you saturate the liver. And if you saturate the liver, it gets to a point that it's toxic. And patients have died from that historically. So we're back to, your treatment of efficacy is flirting with toxicity. So where the industry is going with that one is, if you could evolve these viruses, if you could engineer them to now be specific to a tissue type with higher efficiency and detune for liver, that would increase your likelihood of having a successful drug. And this actually fits at the junction of, AI, RNA, and synthetic biology. So in our case, back to the Alzheimer's story, I need to deliver these RNA to the neurons of patient. The path that people have taken is, intrathecal, so, spinal tap, if you will, to deliver the EV, or intracranial, they drill on your skull and inject the EV. But even if you do that, the EV kind of leaked out of the brain, very inefficient. And, I wouldn't want to sign up to get my skull drilled for a treatment that's highly risky. So what we've been doing is, what if we can mutate these AV in a way where you could give a sample IV injection in your arm, and it would cross your blood-brain barrier and transduce neuron with high efficiency, yet not go in the liver or other tissue. That would be the tool that we need to deliver these RNA and potentially deliver other payloads from other companies. So the way we did this is we studied how these viruses are preferentially binding liver, and we changed the amino acid code the same way we've been doing with RNA. We just brute force our way into making every possible permutation of every amino acid at these locations. What we're trying to do is create random motifs that now recognize neuron instead of liver, instead of hepatocytes in the liver. And there's other company working on this that are seeing great success. From there, the application becomes one of synthetic biology as well, where once you have a potential candidate AV like we have, you could start stacking more tool to it. You could start saying, okay, I want my payload to be expressed more. I want these AV to be less immunogenic. I want them to have a longer half-life in the blood. So you start playing with all the tool of synthetic biology and RNA-based biology to try to really tweak these assets. So that's one application that we're actively pursuing at Shape. The other one is on the manufacturing. We're making these cell line that can produce these drugs at a much, much higher efficiency. Right, so Sarepta's drug, I think they're pricing at 3.3 million per treatment. Right, it's steep, but what's the life of a kid, right, that has this disease? Ultimately, it's tough to be judge and jury of the price of a drug like this. But what I know is that's not sustainable long-term for society, right? I'm trying to cure Alzheimer. There's 5 million patients in the US with Alzheimer. If I charge $1 million per patient, I'm going to bankrupt the economy quicker than COVID. So that's not going to work, right? And maybe I have a Canadian bias towards free healthcare, but I believe that if you change the tool, how we make the drug and how we discover the drug, those $3 million treatment, in a few years, or $300,000 treatment, and a few years later, 30,000, and so on. And theoretically, I do believe gene therapy cost of good could come down to $2 to produce. And I'm not saying the pharma would charge $2. I'm just saying that if it costs $2 for pharma to develop and produce, then we're going to get a more palpable price where we can start thinking about curing kid that has its own disease in the world. Because right now, no pharma are chasing N of 1. That's too costly. It's not worth the R&D effort, right? There's no return. And pharma cannot chase large disease like Alzheimer at scale. It's just not sustainable. So changing how you build a tool to manufacture, how you do the tool for discovery, impacts cost of drug long term. And that happens by doing a lot of AI to shortcut R&D cycle. And it happens by doing synthetic biology so that you change how drugs are made in a much more efficient, cost-affordable way. Ultimately, if you can reduce down a problem to its raw material component, and you hear Elon talks about that all the time about this Tesla battery, but it's the same with life science. COVID vaccine, for example, they cost double-digit dollar, like $10, $20 to produce at best. They made so much of them that they were able to reduce the cost to how much does it cost to buy ribonucleotide to make the RNA, right? And then you could start sourcing the same way you're sourcing cobalt or copper at the lowest cost. This is how we need to bring down the industry of life science down to bearable cost.
Wonderful. Okay, thank you. That was really, really helpful, really insightful response. We've kind of come to the formal end of our discussion here, and at the end of every episode, I like to talk to my guests about who they are when they're not in the office or the lab. And so my question for you is, any good books you have read recently that you would like to recommend to our audience?
Yeah, I've got a lot of books I should read more. I'm getting crushed by my son and my wife there on that front, the avid reader. I recommend this book. I see too many CEO on stage talking about machine learning, and then they get asked.
Machine learning book. Okay. Who's it by?
Yeah, it's by Andrei Burkov. Okay. It's going to make you go back to school in your calculus a little bit. Yeah, you're going to have to do a bit of work. But, just yesterday I was looking at a recording from ESGCT, which is a big conference in gene and cell therapy that happened a month ago. And the CEO is talking about machine learning the whole time. And first question he gets is, are you using regression model or generative model? And the CEO says, I actually will have to refer to my bioinformatician team. And you can't on one end just trying to pick your way into it and not know what you're talking about. I'm not saying like you need to be able to do machine learning, but if you don't know the basis of it, just, don't be that guy. So this book is a good book for people to really get a lot deeper and a very quick 100-page read. It's not easy to read. It's not for most people, but at least then you'll know a bit about what not to talk about. You'll know enough not to say anything stupid. The other book I'm reading actually right now is this bad boy that I've read when I was a kid, The Foundation from Isaac Asimov.
Sure.
This book is quite interesting. It's a brick. It's a trilogy. It talks about this mathematician that could kind of predict the collapse of society, and the conundrum was that if they were to give up quicker on what's happening, yes, it would be horrible quicker, but that duration would only last, some 500 years as opposed to 5,000 years. And he's trying to convince the world to just give out. And obviously, nobody wants to give up. So they get into this, blackout for a lot longer. It's interesting because it's a bit of a vision of that doomsday of AI. Yet at the time, there was no machine learning or AI when Isaac Asimov wrote this, but still based on mathematician prediction of the future. And I feel that's a bit where we're trending with AI, where one day the AI is going to tell us, like, here's what's wrong with what you guys are doing, and here's the solution, and we're not going to like it. It could be like, stop flying, because you're killing this planet.
Right.
And then we're going to have to deal with that reality. So it's a very, very applicable, but today, interestingly, despite not being any AI at the time.
Excellent. Thank you for sharing those. And I fear I may have spoken over you, when you announced the title of the first book. Could you please repeat the title of that first book?
Yeah, the first book was the 100-page machine learning book from Andrei Burkov.
Perfect. Thank you. Well, they both look like excellent reads, and certainly for our audience, I think we will be, perfect recommendations for everyone listening to this podcast. So I thank you very much. All right, listeners, that's it for this episode of Cell and Gene, the podcast featuring Shape Therapeutics, Dr. Francois Nino. And Francois, thank you for tuning in thank you for joining me today. This was a lot of fun and a lot of insight that you provided on AI, RNA, and the intersection between the two. It
was my pleasure, Aaron. Thank you very much.
Good. All right. Well, thanks for tuning in, listeners. Visit us at cellandgene.com, and please recommend Cell and Gene, the podcast, to your colleagues in the space. And we'll talk to you.
Hello, it's nice to virtually e-meet all of you. My name is Dr. Reid Olson, and I am the head of GPCR pharmacology at a biotech company in Oxford, England called Exscientia. The talk that I'm going to be giving today is called The Development and Application of TruePath Biosensors to the Study of GPCR Pharmacology and Functional Selectivity. So before I get started, I just want to thank everyone who worked really hard on the data that I'm about to present to you today. In no particular order, Dr. Brian Crum, a postdoc at UNC Chapel Hill and an amazing structural biologist, Jeff DeBerto, a PhD student at UNC who I worked with during my PhD, Dr. Ryan Strachan, an assistant professor at UNC, as well as a consultant for GPCR drug discovery startups, and a number of postdocs who are now assistant professors at great set of prestigious universities. John McCorvey, who's now a professor at the Medical College of Wisconsin, Justin English, who's now a professor at the University of Utah, and Dr. Tao Che, an assistant professor at the University of Washington, St. Louis, as well as Dr. Brian Roth, my PhD mentor, distinguished professor of pharmacology at UNC Chapel Hill. So I'm very happy to be here today to talk to you a bit about GPCRs and the criticality of robust tools to study their activity with an eye towards high-quality data in drug discovery projects. The main emphasis of this talk will be the exposition of a set of biosensors that I developed during my PhD for this exact purpose, a toolkit that I've dubbed TruePath. I want to emphasize right away up front that these tools are freely available to the academic community worldwide and can be obtained by either contacting Brian Roth's lab at UNC or through direct distribution by AdGene, the nonprofit plasmid repository. So it's very likely that you're aware of the basics of GPCRs and their importance in biology and drug discovery. But just for a refresher, GPCRs are one of the largest protein superfamilies in the human genome. Collectively, they comprise 800 known unique cell surface receptors, the majority of which are olfactory receptors and are key in the mode of chemosensation that we call smell. A large number, however, represent druggable transmembrane proteins that regulate normal and pathological cellular behaviors. GPCRs share a common structure and function. They are cell surface proteins that possess 7 transmembrane spanning domains with an extracellular N-termini and an intracellular carboxy termini. GPCRs transmit information to the intracellular compartment of cells by engaging effector proteins that initiate or alter signal transduction networks, collectively termed transducers. They serve as conduits for the transmission of extracellular stimuli to the cell and integrate all sorts of information, from chemical stimuli to light, even to mechanotransduction. The canonical transduction machinery for GPCRs are heterotrimeric complexes called G proteins, which consist of a G-alpha subunit, which is what we will focus on, as well as both a beta and gamma subunit that function as obligate dimers. The G-alpha subunit consists of 2 major domains, a RAS-like domain which loads different forms of guanine nucleotides, and a helical domain which contains effector binding surfaces for altering downstream signaling cascades. There are additional signaling proteins that alter receptor function or signal on their own, such as arrestins. These are not G proteins, but are still recruited to the receptor upon activation, typically after modification of the receptor by G protein-related kinases. Arrestins function as desensitizers of receptors and mediate internalization. Additionally, they function as scaffold proteins for additional signaling pathways, which can produce unique physiological effects compared to G proteins. The basic activation pathway for GPCRs is through the receptor-mediated exchange of GTP for GDP in the G-alpha subunit. When a receptor is activated, such as through the binding of an agonist, it undergoes structural rearrangements to allow the inactive G protein complex to release GDP and exchange it for GTP. This leads to a dissociation of the subunits, after which they can engage various downstream effectors. Different classes of G proteins have different canonical signaling cascades that they engage. Eventually, the G protein hydrolyzes GTP to GDP and becomes inactive, reforming the heterotrimeric complex with the beta-gamma subunits, able then to engage additional receptors. GPCRs are typically classified by both their endogenous ligand, such as serotonin, as well as the signaling pathways that they are thought to primarily engage. Among the latter, there are 4 major classes defined by the second messenger cascades that they engage. The GI family, for instance, inhibits adenyl cyclases, while the GS family stimulates them. GQ activates PKC signaling, as well as IP3 and DAG accumulation and the release of intracellular calcium. The G12-13 family activates small G proteins, such as ROGAF. There is substantial redundancy within these classes, and the expression patterns of these proteins can vary wildly from cell type to cell type, even when the same GPCR is expressed in those cells. We do not understand the subtleties of the submembers of these classes, and we don't have at present assays that can resolve all of the individual different submembers. But why should you care so much about GPCRs in drug discovery? GPCRs are omnipotent regulators of cellular physiology. Lots of compounds you might be familiar with act through GPCRs. Muscarinic receptor compounds for asthma in the lungs, beta blockers for blood pressure, antipsychotics that act at aminergics in the central nervous system, melatonin receptor agonists for sleep, caffeine for alertness, and plenty of illicit drugs such as LSD, THC, and morphine. GPCRs do a lot of the heavy therapeutic lifting. So if you break down all FDA-approved medications by their protein family class, GPCRs represent the largest group. Despite this, however, the majority of GPCRs in the human genome are untargeted, and the majority of GPCR targeting drugs are clustered in 1 or 2 major families, such as the peptidergic and aminergic class A receptors. So why, despite their success as drug targets, are the majority of GPCRs untargeted? Well, much of the GPCRome is dark. Either orphan receptors where we don't know the endogenous ligand, or a lack of known selective probes to study their function. Frequently, the expression patterns are unknown as well as the signaling pathways that they engage. There's a further wrinkle in that GPCRs can engage different sets of G proteins, both within and between different families. This effect is probe-dependent as well, meaning that different ligands can have different profiles, something that we call functional selectivity. In order to solve both of these puzzles, we need new and improved tools. So what might our options be? As mentioned previously, multiple G proteins share common effectors, so measuring these outputs can obscure which proteins are involved. Another consequence of this is that due to signal amplification and signaling cascades, concepts like efficacy and potency of compounds in these different systems, such as measuring calcium or cyclic AMP separately, are affected depending on the assay as well as the tissue type. A compound with partial efficacy and low potency in one assay may appear as high efficacy and high potency compound in another. So the ideal system would therefore look at something universal among the pathways, so these considerations are more universal. One example of a mechanism that we could look at and exploit is the change of proximity of the heterotrimeric G proteins following activation by an agonist-bound GPCR. So this is an occurrence that is shared among all G proteins and doesn't depend on unique downstream signaling pathways that have different sorts of properties depending on what tissue type you're in or different levels of amplification in the cascade. So one way to measure this sort of thing is with molecular probes, such as fluorophores or luminescent enzymes, which share overlapping excitation and emission properties, such that they can act as proximity sensors through resonance energy transfer. Essentially, if the signal or emission of energy from one donor protein, such as a fluorophore or luminescent enzyme, has resonance with an acceptor protein, such as a compatible fluorescent protein, then you can see and use the emission from the donor to excite the acceptor. Thus, if the proteins are close enough and in the right orientation, you will observe high resonance. When the proteins shift orientation significantly or decrease in proximity, you will observe a decrease in resonance and thus a lower RET ratio. This phenomenon is incredibly sensitive to the proximity of the two proteins, to the point that the signal decays to the 6th power, so it can detect extremely small changes and has very little signal background. The two major approaches to resonance energy transfer are FRET, or fluorescence resonance energy transfer, in which the donor and acceptor probes are fluorescent proteins. Another form is BRET, which instead of using an artificially illuminated donor fluorophore, it uses a luminescent enzyme as a signal donor source. So there are advantages to both of these techniques, such as single-cell FRET versus not having to use a laser to illuminate your donor proteins in the case of BRET. One challenge in getting BRET to scale to industrial drug screening is the sensitivity of the assay, though this is changing with the advent of new enzymes and substrates. The theory for measuring G protein activation is that by putting one half of the RET pair on the G alpha and the other half on the beta gamma complex, we can measure when they are inactive or active based on the ratio of the variable signal of the acceptor to the constant signal of the donor. And in fact, this kind of approach has been used for G proteins before by a number of groups with both FRET and BRET to different levels of success. So only subsets of G protein Brett sensors have been published, and even in the largest subset, the performance of these tools are inconsistent in their quality as biosensors. Here you can see the performance of this toolkit created back in 2012 in Michel Bouvier's lab, a set of Brett sensors based around the design of a set of FRET probes from Paul Devriotti's lab in 2001. In the original design, the donor protein, either a fluorophore or a luminescent enzyme, was inserted at the same position across G proteins in a disordered region in its helical domain. This was due to historical data showing this region amenable to engineering without disrupting protein function, as well as its proximity to the N-termini of the gamma subunit, where they had attached an acceptor fluorophore, in this case a variant of GFP. However, this represented only a partial kit, meaning only some G proteins were included. And while for most G proteins, we were able to replicate signaling functionality with these sensors, the dynamic range of the assay was variable and left a lot to be desired. So the concept, again, is very sound, but we wanted to develop a more complete set of G protein sensors that had also been thoroughly optimized for performance in assays that could then be scaled for drug discovery efforts. So we saw that there were two main challenges, biophysical and biological. As I mentioned just now, the design for each G protein sensor was identical. The luciferase was dropped into the first amino acid at the beginning of the disordered loop between the alpha A and alpha B helices in the helical domain. The conformational diversity of the subunits is also substantial. They move around a lot, especially in their GDP-bound state when proximity is at its highest. When creating resonance pairs, the orientation of the two proteins is also key. One can't just always put the donor or acceptor into equivalent positions of related proteins. We can't just cookie cutter this thing. The luciferase sensor is, for instance, equally sized to the G-alpha subunit. It's massive. Additionally, modifications of the subunits might make them dysfunctional. An ideal probe would be a chimera that is relatively inert, meaning it retains its wild-type functionality. So when it came time to design the probes that ultimately became the toolkit known as TruePath, we wanted to take a brute force approach that combined rational structure-guided constraints as well as exhaustive experimental refinement. We were fairly limited in where to tag the beta or gamma subunits with the acceptor fluorophore, however, which was great, because from early work we found out that tagging the beta subunit reduced trafficking to the membrane with much more fluorescence in intracellular regions. We therefore stuck to tagging gamma subunits with GFP rather than the beta subunit. For inserting the luciferase donor into the alpha, we stuck with the alpha helical domain of the G-alpha subunit because of its proximity to the location of the GFP and its amenability to chimeric engineering. So this is a diagram of where the donor and acceptors were tagged for the GQ protein, where the reference position is the first amino acid in the disordered region between the alpha A and alpha B helices. We've also shown the positions here for the alpha beta alpha C loop. Because orientation and proximity are critical for good Brett probes and their conformational dynamics make purely structure-guided design impossible, we decided instead to target every possible insertion site within those two disordered regions of the loops. Using Gibson assembly, we generated between 12 to 20 chimeric proteins per G protein, ultimately more than 300. In each of these, the luciferase was moved iteratively by one amino acid throughout the disordered loops, all the way from the beginning to the end. So these are diagrams, again, for the design of the GQ sensor indicating the position where the luciferase was inserted into each variant. We then tested each of these chimeras in a dissociation assay using a classically coupled receptor, in this case the neurotransmitter receptor for GQ. As you can see, there is a wide range of performance by the different probe positions. The performance pattern was pretty repeatable, but because of the sheer number of comparisons, we decided to just pick the top 5 for follow-up testing and confirmation, and then development of all of the probes. Ultimately, we chose the best performing chimera as compared to the performance of the reference position, in this case inserting the luciferase at position 126. So now we had an optimal chimera for GQ, which represents 1/3 of the signaling complex, but we wanted to know which beta and gamma subunit set might be ideal for each G protein sensor. So this was a big combinatorial problem. There are 12 gamma subunits in the human genome and 4 primary beta subunits. Testing 56 iterations for each sensor, even after optimizing the alpha, would prove to be too complicated. So we developed a screening cascade for efficient disambiguation of the ideal beta-gamma subunit composition at each sensor. First, we would optimize the G-alpha subunit using a standard beta-gamma pair, in this case beta-1 and gamma-2 because of its use in the original FRET and subsequent Brett probes. Then, we would individually evaluate the performance of this alpha at all 12 gamma subunits. For this step, we co-precipitated the plasmids encoding all of the beta subunits. Subsequently, after identifying the right gamma subunit, we took the best performing alpha-gamma pair and individually tested the performance at each beta subunit. The set with the best dynamic range was then designated as the ideal biosensor composition. So using the GQ sensor from the earlier slide as an example, you can see there's substantial variation depending on the G-gamma subunit, which is used to tag the GFP. In this case, gamma-9 and gamma-1 perform fairly well, especially relative to gamma-2, which is part of the original reference probe set in both the FRET and Brett toolkits. So after determining that the gamma-9 subunit was the ideal acceptor partner, we then compared the performance of the different beta subunits. We sometimes observed more differences than seen here, but in this case, beta-3 slightly outperformed the other beta subunits. To evaluate the performance then that our engineering had, we then performed head-to-head evaluations of the TruePath sensors to the reference set. If a G protein was not among the original set, we used the position that would have been prescribed by the original sort of cookie-cutter design, the first position in the first loop in the helical domain. Here you can see that this combination of rational design and brute force iterative optimization produced a 10-fold increase in the probe performance for GQ. Brings us back to the last biological challenge. We wanted to make sure that we weren't just disrupting the wild-type functionality too much. After all, if the idea is that we are building a tool to study G protein signaling, we want to make sure that they can still signal. So to do that, we employed cell lines that have had different G proteins removed by CRISPR. Here we reconstituted GQ expression using either wild-type GQ or the TruePath biosensor. And as you can see, the performance of both are identical in a classic calcium signaling assay. Targeting the alpha helical domains worked for a majority of the G proteins, but notably there were some subsets for which we were unable to produce functional probes, mostly in the GQ family. G11, G15, and G14, for instance, produced almost no resonance regardless of the insertion site or the beta-gamma subunit composition that we tested it with. This is very odd, as GQ worked great as a sensor, and G11, for instance, is nearly identical in its amino acid composition. But as I have mentioned, the peculiarities of the redundancy of G proteins in families is unknown, and small differences in amino acid sequences can have significant impacts on structure and function. So if the alpha helical targeting strategy wasn't working well for these G proteins, was there an undiscovered area that we could exploit? So to try and solve this issue, we went in search of a unique protein engineering site for inserting the luciferase. We were operating under the hypothesis that either the alpha helical domain was disrupting protein function for whatever reason, or was in an orientation that could not basically produce Brett between the alpha and gamma subunits. So we used homology models of G11 based on a GQ structure because of their near identical amino acid sequence. We explored all residues within 20 angstroms of the N-termini of the gamma subunit. We excluded structured regions and positions within the RAS domain because of its role in G protein activation. We identified a region called switch 3 between the RAS and helical domains to be the ideal region to target in terms of proximity, as well as its putative unstructured state. However, it's been previously identified as an area that was thought to be unamenable to modification with insertions, deletions, and mutagenesis rendering G proteins supposedly unable to signal. Nevertheless, we wanted to test the functionality of these as probes using the same brute force approach as before. In our hands, we found that we were able to reproduce robust biosensors targeting this region when the alpha helical domain failed. We also discovered, in opposition to what the literature suggested, that chimeras targeting this region were still functional and able to activate second messengers. So here you can see in the bottom left that a G11 switch 3 chimera retained wild-type functionality in a classic calcium signaling assay. So this was a very cool and unexpected finding. And ultimately, we were able to extend the strategy to 14 of the 16 non-visual G proteins. We substantially improved the performance of all the probes compared to the previous designs, with a range of improvement from 1.5 to 100-fold and an average improvement of 21-fold. G14, however, never produced any appreciable Brett in any of the constructs. Even replacing the entire helical domain with that of the GQ probe, we were unable to produce a functional probe. It's possible that there's something unique about G14 that we do not understand that precludes generating probes of this design. We also failed to produce a functional G-ULF probe, but we were able to observe a Brett signal, unlike the case of G14. However, no matter which sensor variation or receptor we used, we could not produce a concentration response. We still do not understand why the GS, a related G protein, was able to function as a probe, but not G-ULF. So in conclusion, we demonstrated that in generating Brett probes, that exhaustive design and refinement works, and rational intuition from structures and models only gets you so far. The composition of the beta-gamma subunit matters, not so much for functionality, but for optimal signal. Additionally, the G-alpha subunit is robust to modification even outside the alpha helical domain, a surprising but welcome finding. But we also wanted to demonstrate the utility of the probes. So while the old way of thinking about GPCR signaling is that each one couples to a G protein family, the truth is that they can couple to multiple families, as well as being selective within those families, something called functional selectivity. Thus, the physiology of GPCR activation can potentially get very complex very quickly. So to demonstrate the ability of these biosensors to map available transducer pathways, we selected a set of well-studied and understudied receptors. We then performed what we call transducerome profiling with these receptors against all 14 TruePath probes, using their endogenous ligands. Surprisingly, we found that while most receptors coupled to many G proteins across families, the range of promiscuity was extreme for some and very limited for others. For example, neurotensin seems to couple to nearly every G protein, while the 5-HT7 receptor exclusively engages GS. Notably, the engagement of these pathways in terms of efficacy and potency were different for the same ligand, providing support for the concept of functional selectivity, where compounds at receptors have different preferences for pathway engagement. Looking more closely at a subset of these pathways activated by the beta-2 adrenergic receptor, you can see that it effectively activates its traditional G protein, the GS subunit, as I mentioned previously. It also shows selectivity between the related GI class proteins, activating GI2 but not GI1, something that's been reported in the literature. We also found that it appeared to activate the GQ-like protein G15 with greater potency than its classic G protein, GS, albeit with lower efficacy. But was this just an artifact of the system? I mean, could the receptor actually engage this unreported G protein? So to make sure that these coupling events were real, we employed those G protein knockout cells that I mentioned earlier. We showed that only in the presence of G15 and beta-2 together could we activate G15 in a classical calcium assay using epinephrine. This effect was completely abolished in the presence of alpranolol, a beta-2 receptor antagonist, providing support that yes, the endogenous ligand to epinephrine could signal through the beta-2 adrenergic receptor and activate the G15 pathway. We then applied the TruePath system to profile an entire family of related GPCRs. As a prototypical family, we chose the 5 dopamine receptors, used their shared endogenous ligand dopamine. Classically, D1 and D5 are grouped together as D1-like GS-coupled receptors, while D2, 3, and 4 are classified as D2-like GI-coupled receptors. The reality we observe is that D1 and D5 have actually very little in common aside from GS coupling, with D5 exhibiting a wide range of GI coupling. Similarly, the D2 family was further subdivided, with D2 and 4 sharing nearly identical profiles, and D3 behaving more exclusively coupled to GOA, B, and Z. The consequences of this physiologically is not well known, but you can imagine working in a model cell system that may lack some of these G proteins and concluding that your compound is inactive because you don't observe a response, and that being due to the lack of the appropriate G protein that also, however, may be present in vivo. But what about even deeper profiling? Is there a way that we can use this to start to explore the diversity of in vivo effects downstream of receptor activation at the same receptor? There's growing interest in safer opioids, for instance, and the kappa opioid receptor is a hot target for drug hunting. However, activation of this receptor by different ligands can alternately produce different effects to varying degrees. These include itch reduction and analgesia, but also profound anxiety and intense hallucinations. Theoretically, this could be due to unique signaling profiles by different drugs. Well, we were not prepared to answer the question of what pathways drive these different effects. We wanted to demonstrate that different ligands at the same receptor could engage different G protein families in different ways. In other words, that the engagement of all the G protein pathways were not uniform across all ligands. So here's a fingerprint, if you will, or a transducerome profile of 8 agonists of the kappa opioid receptor. I'm only showing the pathways which produce a measurable response, and you can see that for these compounds, the kappa opioid receptor is very restricted to the GI class of G proteins. Nevertheless, there is different variation across G proteins for the different ligands. So an example of some of the differences that we see are the range of efficacies for its endogenous ligand, dynorphin, from nearly inactive at gusteucin, a taste-sensitive G protein, to partial and nearly full activation at GI3 or GZ, with a range of efficacies between them. So conversely, the hallucinogen salvinorin A almost equally activates most G proteins in terms of efficacy and potency, but is far more active at GZ and much weaker at gusteucin. However, in addition to its application to just studying functional selectivity, the peculiarities of TruePath also allow for the study of more advanced pharmacological concepts, concepts like inverse agonism and allostery, which are not universally available in all signaling assays. So receptors, for instance, can engage signaling pathways even in the absence of an agonist, producing a signal just basally. An inverse agonist turns down this basal signaling level, however. Many assays are not sensitive enough to reveal this behavior or are not designed for it, and so often compounds are classified improperly as inactive or silent antagonists, but in effect they actually do change cellular signaling properties in a cell even in the absence of an agonist. So here you can see the bona fide CB1 receptor inverse agonist produces negative efficacy in a TruePath assay, while an agonist produces positive efficacy. These are the two curves sort of moving in opposite directions. It can also, for instance, be used to study the intricacies of allostery. So allostery refers to the alteration of receptor function by a compound outside of its canonical ligand binding pocket. The interaction of an allosteric compound can alter the behavior of an orthosteric ligand. In other words, it can decrease or increase its affinity and alter its efficacy or have other effects. It's a bit of a rabbit hole to get into deeply for this talk, but allosteric compounds can have significant effects on receptor activity, as well as altering how it can bind the classical ligands. So in this example, you can see that an allosteric modulator here, I've obscured the name just because it's from an upcoming paper, an allosteric modulator of the neurotransmitter receptor alters the activity at the GQ and GI pathways differently, implying that the way the receptor engages the same ligand and different G proteins are in fact substantially different. There's a lot more to the story around this compound here, but it's in an upcoming paper from Brian Crum and Brian Roth's lab. And suffice to say that using TruePath, they're able to show that even G proteins in the same family, so for instance within GI or GQ, can be modulated differently by the same orthosteric and allosteric pair, and that these differences can be teased apart using TruePath, which is pretty cool. Lastly, in terms of applications here, I just want to also mention that the sensors can also be applied to the study of G proteins themselves. So in a paper that was just accepted from Dr. Henrik Dolman's lab, a graduate student named Kevin Knight identified a triad of conserved residues in the G-alpha subunits that are critical for the dissociation of the heterotrimeric complex in activated G proteins. As part of his project, Kevin used mutated TruePath sensors to demonstrate the universality of this mechanism. Additionally, groups are also using this sensor to study the role of other key residues in G protein function. It's been really cool to see how people are applying TruePath creatively to domains outside of like just GPCR signaling and transducerome mapping. The last thing I'll touch on here is the concept of signal amplification that I alluded to early on. So in many classic signaling assays, receptor occupancy is substantially uncoupled from potency. This is frequently due either to massive overexpression of the receptor or multiple steps of transduction pathways with inherent signal amplification, so multiple steps far removed from the initial G protein activation. And what this means is that in recombinant or other cellular systems, you can get substantially different measures of efficacy or potency depending on how well coupled the system is, where you're measuring in the pathway, etc. So when working in drug discovery, where you want maybe potency or efficacy values to be modulated in your SAR, you probably want those to be convertible between pathways, or to have a measure that can be well correlated to the changes in chemical structure, again, in reference to SAR. And you want that to have high fidelity, sort of non-amplified data, which can be really critical. Basically, the TruePath assay it's already well insulated from signal amplification, that it doesn't measure something downstream of an enzymatic cascade. Rather, it's one of the earliest steps in the activation pathway, just the G protein dissociation. However, it could still be sensitive to receptor overexpression, because this is, again, a recombinant system. So in this experiment, we chemically inactivated overexpressed mu-opioid receptor and looked at signaling by the peptide DAMGO through the GI pathway, GI3. In a system which is sensitive to receptor reserve, doing this will actually alter potency before efficacy. You'll see a steady right shift before the efficacy drops down. But in a system that is insensitive to excess receptor, you'd observe a decrease in efficacy before potency, which is what we see here. So this would suggest that what we would expect is actually fairly good concordance between receptor occupancy and potency, and greater faith in our estimates of efficacy. So when we compare the performance of either a classic cyclic AMP accumulation assay or TruePath for the beta-2 adrenergic receptor, we get predictions of sub-picomolar potency in the cyclic AMP accumulation assay, which is downstream of receptor activation in multiple steps in the signaling cascade, or mid-nanomolar potency in the G protein dissociation assay. So in the TruePath assay, the potency estimate actually matches up really nicely with the affinity of isoproterenol, further suggesting that the TruePath assay system provides high-fidelity, clean measures of receptor engagement and can differentiate between measures of occupancy and efficacy. So why is that really useful? After all, classical drug discovery methods have been extremely useful in the past. So an example of this would be in a project that I worked on with the Class B GPCR GLP. I was hoping to understand the mechanism of action of a small molecule clinical candidate compound from Pfizer. So in the same cyclic AMP accumulation assay that I showed earlier, we were seeing that the Pfizer compound was nearly as potent as the endogenous agonist GLP, and with equivalent full efficacy. However, when we were performing radioligand binding competition assays, we saw that its affinity was actually extremely low, very sub-micromolar. Micromolar. So by all accounts, this shouldn't be happening. There should be much better concordance in function if they were both full agonists. So to try and resolve this distinction, we screened the Pfizer compound and the GLP peptide in the TruePath system. And what we observed was rather striking. So the potency of both the peptide and the Pfizer compound matched up very nicely with the affinity from the radioligand binding assays, which you'd kind of guess given the data that I showed you from the mu-opioid receptor experiments. However, the Pfizer compound was substantially more efficacious than the GLP peptide, driving receptor activity 2 to 3 folds greater than the saturating effect of the GLP peptide. So this is an interesting phenomenon called superagonism. The consequence of this is that in the cyclic AMP accumulation assay, which is heavily amplified, it's like turning up your speakers so loud that you lose the ability to differentiate between some frequencies. It overwhelms your microphone, imposing a ceiling. Everything just kind of gets flat. So the curve then, when it hits the top and you can't detect changes in efficacy, basically sort of shifts to the left, so that the efficacy translates into enhanced potency. And you could see that if you're trying to sort of develop a compound using SAR and you want to maybe make something more potent, but maybe you want to change the efficacy in a different way, being able to differentiate those measures and correlate them with changes in chemical substituents on your scaffold can be pretty key. So in conclusion, just to sum up what I've presented here, a little bit about TruePath. The most completed set of G protein sensors to date, robust, well-optimized, functionally validated. It can be used to identify previously unknown signaling pathways downstream of receptor activation. You can use it to profile different compounds to identify functional selectivity. It can be used to study inverse agonism and allostery, and non-amplified systems like TruePath provide separate approximate measures of receptor occupancy and efficacy, which can be really key in strong SAR. And I really, really want to emphasize this is open, freely accessible resource for the academic community. We made it to solve complex problems, and it only works if other people find a use for it. So we're hoping that people find this useful, people reach out, people get it, test it in their labs. If you have any questions about it, I'm happy to take them now, happy to be contacted by email. If you're using this in your lab and you have any questions, please contact me. I'm happy to help you guys troubleshoot. It was a great project to work on, and I really, really enjoyed it, and I hope that you find it useful as well in your work. I just want to acknowledgements again, Brian Ross Lab, everybody I mentioned previously, UNC, Henrik Dolman, Kevin Knight and Natalie Valentine, 2 grad students that I mentioned, as well as my colleagues in the GPCR team at Exentia, Dr. Katie Smart, Dr. Alice Barizzi, and Dr. Laura Thompson. My funding sources, UNC, Thomas Collin Butler Fellowship, and NIH, National Institutes of Neurological Disorders and Stroke Training Fellowship. So again, thank you for your time, and I hope you enjoyed the talk.